Hello again, everyone, and welcome to Are in Pharma. Today, it is my great pleasure to introduce Dionne Price, our first keynote speaker. Dionne Price is the Deputy Director of the Office of Biostatistics in the Office of Translational Science in the Center for Drug Evaluation and Research at FDA. In this role, Dr. Price provides leadership to statisticians involved in the development and application of methodology used in the regulation of drug products. She currently leads cross-cutting collaborative efforts across FDA to advance and facilitate the use of innovative trial designs in pharmaceutical drug development. Dr. Price received her master's in science in biostatistics from the University of North Carolina, Chapel Hill, and a PhD in biostatistics from Emory University. Dr. Price is an active member of the American Statistical Association, ASA, and INAR, the Eastern North American region of the International Biometric Society. She's a fellow of the ASA and the 2023 president of the ASA, and today she's going to talk about our impact in the evolving data landscape. Dionne, the floor is yours.
Thank you so much, Elena, for that very kind introduction. And good morning, everyone, and welcome to the R in Pharma conference. I would like to first thank the organizers for the invitation. It is indeed my honor to speak to you all today. And as you've heard, I've entitled my talk, Our Impact in the Evolving Data Landscape. And this is my standard FDA disclaimer. So for the time we have together, I will first introduce what I mean when I say the evolving landscape. My perspective is, of course, shaped by my daily work at the FDA. So I will share a bit about the FDA and then discuss challenges which also represent opportunities and briefly touch on the evolution of R. First, there have been important developments in statistical ideas based on an evolution of science and problems. Andrew Gelman and Aki Vatari have a 2021 paper on what are the most important statistical ideas in the past 50 years. Some of the most important ideas include counterfactual causal inference, bootstrapping and simulation based inference, over parameterized models, robust inference, genetic computation algorithms, adaptive decision analysis, and exploratory data analysis. The paper ends with a section on looking forward, and I quote, "What are the biggest challenges and opportunities facing statisticians? Three related trends and applications are big data, messy data, and complicated questions. In some ways, these trends go together. When using data from more sources, it should be possible to make more finely grained inferences and decisions in problems ranging from personalized medicine to recommender systems to robot cars." End quote. And over the years, our sources of data have evolved. For example, many of you probably have on a watch, such as I do, that can collect data, and you may use that data to inform a health decision or other decisions. I know my watch tells me that I need to stand up every so often. And if we think about the pandemic, there were massive amounts of data from electronic health records and other sources. Statisticians and data scientists were front and center throughout the pandemic, using data to inform decisions about potential treatments and vaccines for COVID-19. Climate change is another area where we have massive amounts of data, and we can use that data to impact and inform decisions. There is an area, this is an area where statistical research and computational prowess is needed to assist in managing and mitigating the impact of climate change. Machine learning is another evolving topic where we as statisticians and data scientists should provide leadership and be at the table. Items over the last few years in print have been quite interesting, including a December 2022 article, I believe, in the New York Times that reported on facial recognition technology being used by Madison Square Garden to ensure banned patrons are not allowed to enter. In February, and even more recently, several news outlets have reported on ChatGPT, an artificial intelligence chat box. It supposedly has the capability of performing professional tasks such as writing software code and preparing documents. Artificial intelligence is being used in healthcare, as evidenced by this article in the Journal of the American Medical Association on development and validation of a deep learning algorithm for detecting diabetic retinopathy in retinal fundus photographs. And closer to home for me, AI is also being used and seen at the FDA, as evidenced in an app that recognizes strokes and approval of a machine learning application. Now, these apps were likely reviewed by our Center for Devices and Radiological Health, which is a different center from where I work, which is a perfect segue into the FDA structure. As I'm sure most of you know, the FDA is made up of multiple centers, including the Center for Biologics Evaluation and Research, and they focus on vaccines and gene therapies, for example. The Center for Devices and Radiological Health, the Center for Drug Evaluation and Research, which is where I work, the Center for Food Safety and Nutrition, the Center for Tobacco Products, the Center for Veterinary Medicine, and the National Center for Toxicological Research. We all use data and all have quantitative thinkers such as statisticians and data science. And each center has an organizational structure. And we generally in the federal government use a lot of acronyms, so FDA is no different. We refer to the Center for Drug Evaluation and Research as CDER. We have super offices within CDER, including the Office of New Drugs, the Office of Surveillance and Epidemiology, the Office of Translational Sciences, and the Office of Generic Drugs. And our Office of Biostatistics is housed in the Office of Translational Sciences. Now, this is our organizational structure for the Office of Biostatistics. We have 10 divisions. Our division of Analytics and Informatics is our newest division, and this is where our master's level statisticians are housed, and they provide superb analytic and computational support to the divisions of Biometrics 1 through 9. Our divisions of biometrics 1 through 5 and 9 all focus on pre market drug evaluation before the drug is approved. And our divisions of biometrics 6 is a bit of a specialized division. They work with our pharmacology and toxicology colleagues, so they may be looking at the stability of a drug or the if a drug is carcinogenic. They work with our chemistry and biotech colleagues as well as our controlled substance staff to ensure that new opioids are abuse deterrent and cardiac safety studies. Our Division of Biometrics 7 primarily focus on post-marketing safety. So once the drug is out there, it's been approved, we may start to see safety signals that we did not see in the controlled clinical trial environment. So think, large observational studies. And then our Division of Biometrics 8 focuses, excuse me, on generics. And I should say our divisions of biometrics 1 through 5 and 9 are all divided by therapeutic areas. And each therapeutic area has its own unique set of challenges. Now, I never like to show an org chart without talking about our mission. So, our mission in the Office of Biostatistics is to provide CEDAR and other stakeholders with the statistical leadership, expertise, and advice to foster, of course, the expeditious development of safe and effective drugs and therapeutic biologics. And we protect the public health by applying statistical approaches for monitoring the effectiveness and safety of marketed drugs and therapeutic biologic products. Now, in carrying out our mission, we often encounter challenges, which in my mind, challenges represent opportunities. So, some of the challenges are represented as topics in the seventh iteration of the Prescription Drug User Fee Act, better known as PDUFA 7. The document is available online, and I draw your attention to a sentence in the beginning of the document, which states the performance and procedural goals and other commitments specified in this letter apply to aspects of the drug review program that are important for facilitating timely access to safe, effective, and innovative new medicines for patients. This will allow FDA and the regulated industry to identify emerging challenges and develop strategies to address these challenges to ensure efficiency and effectiveness of the human drug review program. Some of the topics in PDUFA 7 include rare diseases, real world evidence, digital health technologies, patient focused drug development, and complex innovative trial designs. This just represents a few topics that I pulled out, but there are others. For example, model informed drug development. These topics are not mutually exclusive. And today, I will just briefly focus on rare diseases, digital health technologies, and complex innovative trial designs. So let's quickly look at rare diseases. There are numerous challenges in rare disease drug development. For example, the natural history of rare diseases is often poorly understood. These are diseases that are progressive, they're serious, life limiting, and often lack adequate approved therapies. They're very small populations, and that small population impacts our study design options. There's heterogeneity within a disorder or a rare disease. Drug development tools are often lacking. There's often a lack of precedent in terms of what's a clinically meaningful endpoint. And there are ethical considerations for children in clinical trials, and there are quite a few rare diseases that impact children. Now, with these challenges, as I've said, come opportunities for statistical innovation and a need for computing and graphical capabilities. Some of the opportunities include incorporation of external information into trials in a Bayesian framework. And some of the external information may come from patient registries or early phase trials or natural history studies. If you're in a Bayesian framework, the choice of priors and the decision criteria via posterior distributions become important. Other opportunities include small n sequential multiple assignment randomized trials. Kelly Kidwell at the University of Michigan has been a leader in this Essence Smart designs. Adaptive designs with multiple adaptations, N of one designs, randomized withdrawal designs, platform trials, and analyses of multi component endpoints. CEDAR is committed to our accelerating rare diseases. So, with that, we have developed ARC, or Accelerating Rare Disease Cures Program. And the vision of the program is to speed and increase the development of effective and safe treatment options addressing unmet needs of patients with rare diseases. And more information is about the ARC program is on the website, externally available. Rare diseases is an area ripe for innovation, which is a nice segue into complex innovative trial designs. Before, say, 2017, which would be pre PDUFA 6, um, we noticed that there was a limited use of innovative trial designs to provide substantial evidence of effectiveness across a broad range of therapeutic areas. So we may see innovative designs in the exploratory space, but we didn't necessarily see it those designs to in the substantial evidence of effectiveness space. There was a lack of clarity around regulatory acceptance and guidance of some of the innovative trials, lack of experience and understanding. The FDA could not discuss innovative trial proposals under investigational new drugs. As you all know, we can only discuss publicly available information, which usually becomes available once we have approved a drug. Despite all of this, there was a realization of the promise of complex innovative trial designs, commonly referred to now as CIDs. Thus, we started the Complex Innovative Trial Design Pilot Program, and that was under PDUFA 6, but the program is being continued under PDUFA 7. The program is intended for highly innovative trial designs with a particular focus on designs where simulations are necessary to evaluate the trial operating characteristics. Again, an opportunity, as these simulations are often complex and require extensive computing and graphical capabilities. Now, I often get the question, what exactly is a CID? So, in our guidance, interacting with the FDA on complex innovative trial designs, we actually say there is no fixed definition because what is considered innovative or novel today can change over time. So, for the purposes of most of the work we've been doing, we say that CID includes trial designs that have rarely or never been used to provide, again, substantial evidence of effectiveness in new drug applications or biologic licensing applications. So, let me tell you a little bit about the Complex Innovative Designs Paired Meeting Program. Again, it's being continued under PDUFA 7. It's a joint effort between the Center for Drug Evaluation and Research and the Center for Biologic Evaluation and Research. Sponsors submit designs through meeting requests, and if accepted, they have the opportunity to engage with the regulatory team on designs via two meetings. And I stress regulatory team because this program is actually led by the Office of Biostatistics, but it cannot be successful without our colleagues, our multidisciplinary colleagues. The agency selects up to two submissions per quarter, and this was a very innovative part of the program when it started under PDUFA 6. We, in turn, get to use the designs as case studies for continuing education and information sharing. So, what that means is at the IND stage, the FDA can speak about these innovative designs. And we have an agreement, a disclosure agreement with the companies to ensure that the elements that are being disclosed, we have the company's okay on those elements. And there's a 5 year duration. So there's a 5 year duration for the pilot, and then there's a 5 year duration for the continuation of the peer meeting program. So there is no set definition, as I mentioned, but we do list some features of CID. So, for example, you could have complex adaptive designs, and these could be seamless designs where you're combining dose finding and hypothesis confirmation in a trial. You could have multiple adaptations, such as you're adapting the treatment arm selection, the patient allocation, and/or the endpoint selection. A formal incorporation of prior information, and we usually think of this in a Bayesian paradigm. So, for example, if you have a placebo arm, you may augment it using external controls or other data sources. And again, this is where some of that overlap comes in because the other data sources could be real world data. Leveraging or borrowing strength from information internal or external to the trial. Use of a posterior probability to determine trial success criteria. And again, this would be in a Bayesian framework. Master protocols, which encompasses platform trials, umbrella trials, basket designs. So, for example, platform trial, you may have multiple companies investigating their individual drugs, but it's under one protocol. Excuse me. Excuse me. And we have a shared control arm. We saw an increase in interest and use of master protocols during COVID 19. Again, that represents an opportunity for scientific thinking as well as computational and graphical capabilities. And then sequentially, multiple assignment randomized trial smart designs. So, as a FYI, under the pilot program, I believe we accepted 6 submissions, and the case examples are on the FDA CID website. And of note, simulations from some sponsors and within FDA were conducted using R. Same with COVID and those master protocols. Some of that work was also conducted using R. So, with all these new data sources, it's no surprise that digital health technology has come front and center in drug development. And we first start with what is digital health technology. So, this is a definition taken from the FDA NIH Best Glossary, and it says a system that uses computing platforms, connectivity, software, and sensors for healthcare and related uses. And these technologies span a wide range of uses from applications in general wellness to applications as a medical device, and it goes on. Examples of DHTs include wearable, implantable, or ingestible sensors such as accelerometers, continuous glucose monitors, heart rate monitors, software applications, apps that may collect clinical outcome assessments, and other general purpose hardware, say a mobile phone camera. Under PDUFA 7, the FDA has made several commitments around the topic of digital health technology, including developing a DHT framework, establishing a DHT steering committee, convening a number of public meetings and workshops, identifying demonstration projects, developing a guidance on the use of DHTs in traditional and decentralized clinical trials, developing prescription drug use related software guidance, expanding our review capacity and expertise, and enhancing our IT capabilities. We do have a draft guidance on digital health technologies, but it's specifically on digital health technologies for remote data acquisition in clinical investigations. So I've mentioned or presented some topic areas, again, not an exhaustive list, that represent opportunities to advance information both through scientific thinking as well as computing. And these include complex innovative design, our paired meeting program, and we will be issuing a guidance on Bayesian methods, and we will have a workshop in March of next year. The model informed drug development paired meeting program, accelerating rare disease cures, or our ARC program, advancing the real world evidence pilot, and then enhancing the use of digital health technologies. And all of these come under PDUFA 7. So now I'm going to date myself just a little bit. I have been at the FDA over 20 years. When I first started, I was straight out of grad school where I was proficient in SAS and S plus. And if requested, I could program in Fortran and C. When I started at FDA, I primarily used SAS. However, as I mentioned, that was over 20 years ago. Today, our employees are proficient in SAS, R, and Python. And given this evolution, FDA issued a statement in May of 2015 clarifying that FDA does not require use of any special software for statistical analyses, and statistical software is not explicitly discussed in the Code of Regulations. It goes on to say, however, the software packages used for analyses should be fully documented in the submission, including version and build identification. And then the statement goes on. This is the statement in its entirety. So, with this statement, I propose that there's been an increased interest in R-based submissions. And as you all well know, November of 2021, the R Consortium announced submission of an R-based submission via a pilot, and it was called Pilot 1. The objective of the pilot was to test the concept that an R-based submission package could meet the needs and expectations of FDA reviewers, including assessing code review and analysis reproducibility. The components of the submission included ATOM data sets and XPT files, a PDF report with analysis outputs, the analysis data reviewer's guide, R programs, and a proprietary R package. The FDA was able to receive the electronic submission package in eCTD format, reconstruct and load the submitted proprietary R package, install and load open source packages used in the submission, and reproduce the analysis results. And there's a pilot 2. The objective of Pilot 2 was to test the concept that a Shiny application created with the R language could be bundled into a submission package and transferred successfully to FDA reviewers. The application was built using the source data sets and analyses contained from Pilot 1, and the FDA review was issued in August of this year and is available online. So I hope you would agree with me in saying, with the changing scientific and data landscape, challenges and opportunities prevail. And I always say a challenge represents an opportunity. There will be an increased need for computing and graphical capabilities. And I hope you enjoy the conference. Thank you for your attention.
Thank you. Thank you, Dionne. We are going on a 10-minute break now.
Okay, thank you. Have a great day.
Thank you. we have time for questions. Okay, we do have time for questions. So is Dionne still here?
I'm
Yes.
still here. I
Okay,
was
thank
taking
you.
a sip of water because I was trying my best not to cough, but I have a cough that I've been trying to hold as I was talking.
Okay, so where are the questions? Are they in the comments area or?
I do see one question. It's one I actually don't have a good answer for. Someone is asking, how do we ensure reproducibility when it comes to using complex randomization with priors using Bayesian analysis, such as STAN? It's a great question, and one that certainly is at the forefront of our minds in terms of reproducibility. But you ask, how do we ensure? And I think we're still working on that aspect of how do we ensure the reproducibility. We do work with the companies and ask a lot of questions. Usually there's some type of kind of analysis, report and a plan. So we work with companies to do our best to ensure reproducibility. But it's a question I don't know that I have a great answer for. I see another question. Are you aware of agencies from other countries, including the EU, which have had our submissions? I am not aware, but that does not mean it has not occurred. Usually, what we see, I believe other agencies see as well. So my guess is they're receiving them or seeing them as well.
Do we have other questions? Okay, I think this is all for the questions. Thank you again, Dionne, and let's go on a break. We have a 10-minute break now.
Okay. Thank you again for the invitation and have a great conference.
Thanks.